Thanks. So while we're going to try to fix the mouse, because I know Dan has incredible graphics in his slides, I think we all know Dan, reading through his background, knowing him for over 20 years, but reading through his background, he has that Harvard pedigree. He's led the breast cancer programs at Dana-Farber, Georgetown, and University of Michigan, all top-notch institutes with top-notch oncology programs. I want to go back to some things that I've observed in his career, such as Dan has been the person who has made biomarkers just as important as drugs. And he was ahead of the curve when he started talking about biomarkers. And Dan, as I recall, you led a National Academy of Sciences committee or report on the use of biomarkers. And I'm probably misquoting you, but paraphrasing and saying that you coined the phrase that a bad biomarker is just as bad as a bad drug. And I've always remembered that, and I think that every trial that we put together should include a priori biomarkers, and I credit opening up the field to you. I also want to credit you, Dan, for ASCO presidency takes a little bit of time. The year before, the year of, and the year after, and I've noted that at the last couple of meetings, yet you stayed on as breast TM chair. So your commitment to SWOG, to clinical trials and the patients is magnificent, and we all thank you for that. And with that, I'll have you come up here and tell us about what you've learned and what you can teach us from 25 years of SWOG breast TM chair. Dan requested Hey Jude be played during his introduction.
To advance and then the mouse to point. The thing is, it's if you move it too much, it'll go on the screen, and it's hard to find.
Okay.
So yeah, and there's a laser pointer on here too if you need it. It's,
All right. Thanks, Lee. First of all, it warms my heart to see this room so full. Plenary session, translational medicine. Trust me, we started a similar committee in CLGB back in 1993, I think. And the two of you might have been there, and everybody else was gone. So it's terrific to see everybody getting religion on this. I'm going to go through some history, and I apologize in advance. I've almost certainly left somebody out of this, and you're going to come up to me afterwards and say, well, I did this, or so-and-so did that, and I'll say, yes, Lee would only give me an hour and a half for my talk, so. To start with, I actually do have a conflict of interest related to this, and that's related to CTCs. I've transparently told several of you about it several times, but University of Michigan, you'll notice I'm wearing my maize and blue tie today, particularly proud of that, has a patent related to the use of CTCs. This has been licensed to the current manufacturer of cell search, and I receive royalties on an annual basis for this. I want to start out with this first slide. We had a strategic planning retreat in 2010. Many of you were there, sort of gaze at our navel, and at least we weren't asked to blindfold ourselves and fall over backwards and have somebody catch us, but it was a similar sort of thing. And one thing we were asked to do is go around and write what we thought would be big ideas that SWOG could do. And this was Jimmy Ray's contribution. You'll notice at the very top, number one was get Dan Hayes thrown out of SWOG. Now, I took that seriously, but it took 8 years for that to happen, as I give my swan song here. What is translational medicine? I mean, I really think the field of translational medicine in solid tumors started with the late Bill McGuire and Mark Lippmann when they began to apply the concepts that Elwood Jensen had discovered about estrogen receptor to the clinic and showed indeed that we now use routinely that estrogen receptor negative cancers do not respond to endocrine therapy, and therefore we're quite comfortable not using it. So what is translational? It's lab to clinic, clinic to lab, therapeutics or diagnostics or both, but in my opinion, it's that for those of us who wear white coats and see patients, at the end of every clinic day, there should be a question that needs to be asked. You should walk out of your clinic saying, why did that happen? What happened to that patient? Dr. Tuveson's talk he just gave. Why don't pancreas cancers respond to the things we have? And if you weren't inspired, then I'm not going to inspire you either. I just thought that was a wonderful talk. But at the end of every research day, there should be a clinical experiment. And I ask my postdocs, my techs, Jimmy Ray, on a weekly basis, so what? If you've shown me data at the lab meetings, so what? What are we going to do with this? How do we apply this back to the clinic? And I think if we keep asking ourselves that way, we'll make headway. Now, there are several reasons to do tumor biomarker studies. One of those is to change practice. And as Lee pointed out earlier, the Correlative Science Committee is designed to give us specimens so we can change practice. The second, though, is to still generate hypotheses for future studies. Certainly there's room for that. Chaos is good as long as we're developing the chaos into organized ways to change practice. The third is to identify unique biologies that might lead to new ideas from lab to clinic. But the fourth, and I think probably the most common reason to do this, is to get published, promoted, or funded, which has been unkindly termed stamp collecting to me in my earlier career, and I've always kept that in mind. What we're trying to do is change practice, not to make pretty pictures. So there are other reasons to do this. One is to test the test. Do we have a test that could change practice? Or the other is, do we have a test that could be used to make a new drug or an old drug better? And these are both things to do. So what does it take to get these into these settings? And many of you have seen these data, but I think they were game changers, published about 10 years ago, are these concepts by Steve Teutsch and the Evaluation of Genomic Applications in Practice and Prevention, EGAP, which was really developed for germline DNA testing, but has applied to tumor markers in general, in my opinion, and adopted by the Institute of Medicine committee to which Lee referred earlier. The first of these is analytic validity. Is it a good test? Does it really identify with high sensitivity and specificity and accuracy what you think it does? The second is clinical or biological validity, and does that test take one group and split it into two or more groups that are either different biologically or different clinically, more importantly? That doesn't mean those of us who wear white coats should use it, but if it doesn't do that, it probably is not going to have clinical utility, and I know you all can read, but I'm going to read the last thing. Do the results of the assay lead to a clinical decision that's been shown with high levels of evidence to improve outcomes? Exactly the same bar we use to apply new drugs in the clinic. We should also use the same criteria to apply new markers in the clinic, because we're using these to direct patient care. So how do we generate these high levels of evidence for clinical utility? And again, this is the Institute of Medicine committee. It was developed for another reason, for the fiasco at Duke, but it was a worthwhile effort to begin to organize how we do tumor biomarkers. On your far left is a discovery phase and the test validation phase to get analytic validity, but more importantly on the right are ways to get high levels of evidence. There are two of those. One is to run a prospective clinical trial, either as a registry or, more importantly, that the marker actually directs patient care within the trial to prove that it's worth using. There are very few of these strategies to test the test, and if you're really interested in this, and if you're in this field, you have to read Dan Sargent's paper, the late Dan Sargent, we all miss him dearly, that he published in JCO in 2005, and Boris Friedland and Lisa McShane published a very similar paper in JNCI in 2010, and these have a variety of ways to test the test. At present, there are very few such trials going on in North America or in the world, but for example, in breast cancer, there are 4 that I'm aware of. I want to point out that 2 of those have been run by SWOG, and I'm very proud of that. The third one, TaylorRx, SWOG was a big contributor to it. So we've been running along pretty well doing prospective trials to test the test. But is that always necessary? Well, no. And I was really honored to be invited by Rich Simon of the Simon two-step hypothesis trial, the phase 2 trial of the Simon and Norton hypothesis, and so on and so forth, to write a paper saying that maybe you don't always have to do a prospective trial, and what you can use is archived tissues. And in this paper, Rich coined the term prospective retrospective studies, and I like that. What it means is you're not just pulling samples out of the freezer, running an assay, and hope you get lucky, which is how many of our studies have been done. But rather, what you're doing is prospectively sitting down and saying, okay, we have archived samples from a prospective trial that ask an important question. Can we use these samples to come up with a tumor biomarker test that actually changes the way we practice? And so that's gained a lot of traction. The issue is, what are the criteria for a good prospective retrospective study? And these are still under regard. But it really raises what is clinical utility, and that requires clinical judgment for the situation. I'm not here to tell you what clinical utility is. You need to tell yourself, and your patient needs to decide. But for example, you'd still have to speak the language. One of my other favorite sayings is, I want you to think like a scientist and I want you to think like a doctor. When I say so what, I also say, is this important enough as a doctor that you'd pull the trigger and use this assay to withhold or to treat a patient with a new therapy? And if you're going to do that, then you have to think like a scientist. How do we get the evidence to get there? This is a study that was published quite a while ago by Alan Coates and John Symes, in which they asked patients who received adjuvant CMF, would they do it again for various levels of benefit in terms of extra survival. For example, if you went from a 50% chance of survival to a 75%, a 25% increase, almost everybody said, yeah, I'd do that again. How about if you go from a 90% to a 95%? Yeah, maybe 3/4 said they would. How about if you went from a 98 to a 99%? Surprisingly, in this study, half the people said they'd do it again. Now, there are problems with this study, but the real point here is you go right to left. An increasing number of patients say, I wouldn't do it again. And the question, are you here, are you here, or are you here? Which of these is of clinical utility? I can't tell you that. I know where I am. I'm in that middle purple in terms of the recommendations I make to my patients, but these are going to differ between diseases, among patients, among countries, and resources. These are all important issues when we're talking about clinical utility. But the power of the cooperative groups to do this lies in the opportunity to take advantage of the clinical question that was asked in the trial. Or even if not, even if we have a trial that doesn't have a benefit of one arm versus the other, we have prospectively collected clinical data, really high quality clinical data. These aren't just samples that were stored in a freezer and you pulled them out and sent a med student to review the charts. These are really important and special samples, and we have the resources to do it right. Now, to my knowledge, these are two of the first tumor marker studies, correlative science studies, translational medicine studies. We keep changing the name, I think, to protect the innocent. But on the far left was a paper by Ed Fisher, Bernie Fisher's brother. Many of you recall Ed is a pathologist, and Bernie, of course, was the surgeon who started the NSABP. And they published a whole series of studies about pathologic findings from protocol B04. And on your right is a leukemia study led by Clara Bloomfield and the CLGB in 1989. I'm sure there are other studies that I've not put in. But then I want to go to some cooperative group studies I was involved with. On your left is a paper we published out of CLGB looking at MUC1 expression in breast cancers and prognosis. You'll notice that they are different. Disease-free survival and overall survival for those who were high versus those who were low, with a p-value that just made statistical significance, so I rushed it off to JCO. It didn't hurt that George Canellis was my boss and he was the editor at the time. Didn't change practice in any way. It did help me get promoted. It's a nice stamp collection, I think, on the left. On your right, though, I began to think maybe we should really ask a clinical question instead of just hoping we get lucky. And this was a study out of CLGB 9344 in which patients received AC and then were randomly assigned to Taxol or paclitaxel or not in the adjuvant setting. And this suggested that patients who were ER positive and HER2 negative got no benefit from adding paclitaxel. That's on the left. And on the right, if they were HER2 positive, they got substantial benefit. This didn't change practice, but it got us to thinking about trials to select chemotherapy based on biology and not just whether or not you had positive nodes or how big your cancer was. Then I made a critical decision in my life. I left Harvard and Dana-Farber and went to Georgetown, and then I left there and went to the University of Michigan. I tried to talk Dr. Baker into moving the entire University of Michigan into the CLGB, and he said that wasn't going to happen based on a single person. And so I joined the Southwest Oncology Group, and it's been an unbelievably lovely experience for me. Chuck Coltman welcomed me with open arms. Bob Livingston then was the breast committee chair and could not have been more gracious. We all miss him too, of course. And Peter Ravden, to his credit, was already doing what I thought I would do. But again, it opened up into taking me into the group and was very open about that. He then left and went on to do other things, and that's when I got to take over the committee. These are the goals of the breast committee from a site visit in 2008. They're just as you'd expect, to start doing what we do and to bring biology into our therapeutic trials. And we designed this algorithm, which is the breast committee led by Gabriel at this time, and Julie Graylow would develop clinically interesting data, design a new trial, generate more clinical data from that trial. We would have specimens available, perform the assays, generate biological data that would inform the next new trial. And this is a cycle that we've been trying to do, and so have many of you, and I think it's a very helpful thing. Now, there are 3 of these I just want to point out. We've done a lot of other things, but these 3 trials are ones I'm particularly proud of and that I think are beginning to pay off for us. The first of these is bringing the 21-gene assay to guide chemotherapy use. So rather than just ER and just HER2, as we did in 9344, could we do better? Working with Kathy Albane, who of course ran SWOG 8814, in which patients were randomly assigned to tamoxifen, CAF with concurrent TAM or CAF followed by TAM. And this was a positive study, but not by very much. We had tissues from the group that got TAM and the group that got CAF followed by TAM. The middle group was confounded, so we didn't do that, and we ran the 21-gene recurrence score. And these are the data. They look very similar to what the NSVP had already published for node-negative patients, but these were all node-positive patients. And what you can see as you go left to right on the recurrence score, that the separation between TAM only and TAM plus CAF gets bigger and bigger, suggesting it's not just prognostic, but it may be predictive. And it didn't matter if you had 1 to 3 nodes or if you had 4 or more nodes, the same picture was seen. It's just your prognosis is worse if you had positive nodes. So anatomic prognosis does still count, but the suggestion was that we could predict who would not benefit from adjuvant chemotherapy based on resistance and not just on whether they needed it. So we ask ourselves, should we change practice? These are data from the Oxford Overview. We asked them to run the samples or the patients in every which way we could figure out. And as you can see here, in every case, no matter whether patients were ER positive, ER negative, young, old, level of ER, grade 3, grade 1, 2, adjuvant chemotherapy reduced the odds of recurrence and death by about 1/3 with no subset effect. So this is probably my most important slide. You'll notice the certainty I have about any issue and the likelihood I am wrong has an R-squared of 0.99. And I've asked Bill Barlow to run the statistics. The p-value is 10 to about the -10th. The point here is that every time I think I know what I'm doing, I get an answer that doesn't make sense. And as Kyle Busch used to say, that's racing. Well, that's science, and that's what we have to accept. So we developed a new prospective study. Kevin Kalinsky is the PI for that, the responder trial for patients who have 1 to 3 positive nodes who are hormone receptor positive and HER2 negative, and who have a recurrence score less than 25, and those patients are randomly assigned to chemotherapy plus endocrine therapy or endocrine therapy only. That trial is completely accrued as of October 2015, and we're waiting very anxiously and patiently for the results to become sufficiently mature to see if this hypothesis is correct. A second group are studies of homologous recombination, or BRCA-ness, in triple-negative breast cancer. Priyanka Sharma got samples from S9313, which has been a real workhorse for us. S9313, as you recall, was basically a randomization of AC versus AC. So the results are identical, as you see on the right-hand side. But we have samples from 2,000 patients, thanks to Peggy Porter. Many of those are in the TMA. And we've been able to rattle off a number of studies with this. Priyanka pulled out all the triple-negative cancers and has done genomic DNA and RNA and everything up which way and out. And now we've begun to see some publications from this. Priyanka published this in the Annals of Oncology just in the last few months. And as you can see here, if you have DNA damage response deficiency, DDRD, you do better in those patients who got chemotherapy. They all got AC and were followed uniformly, than those patients who did not have DDRD, which is exactly what you'd expect. If you can't repair DNA, you're more susceptible to chemotherapy. But so what? This is nice stamp collecting. Well, the so what then is with Eve Rodler. Priyanka has developed a trial that we opened a few years ago, S1416, in which patients who either had germline BRCA mutations known or were triple negative and were known to have germline BRCA wild type. 2 different cohorts, same protocol. Everybody got cisplatin, randomly assigned to Vilaparib, a PARP inhibitor or not, but 2 separately powered cohorts within that study. And when we get all this, we're collecting everything we can get our hands on, and we will be doing a number of the studies that Priyanka has already shown looked important in Protocol 9313, and now we know we can apply them to this study. This study is almost completely accrued with just, I think, 18 months of accrual. It's accrued like crazy, as we expected it would. And then finally, CTCs, and that is to start with clinical trial SO500. This was based on a study several of us did run by the company back in the early 2000s, first published by Massimo Cristofanelli and the rest of us in the New England Journal, showing that if you still had CTCs after a single cycle of some kind of therapy, you're in trouble. It's the equivalent of a path CR, in my opinion, with neoadjuvant chemotherapy. So we generated SWOG SO500 to see if maybe if you chose the wrong chemotherapy as first-line chemotherapy for metastatic disease, if you switched to a different chemotherapy, those patients would do better. This trial was PI'd by my colleague from the University of Michigan, Jeff Smiraj, and this was the trial design. And basically, half the patients out of 624 starting first-line chemotherapy had elevated CTCs to begin with, and half of those did not have a reduction after 1 cycle of chemotherapy to less than 5, and they were randomly assigned to change therapy or stay what they're on. Now, there are 2 important outcomes of this. The first of these is that the patients fell into 3 groups. Arm A, those who didn't have cells at baseline. Arm B, those who did and had a CTC response. And Arm C, those who did and did not have what we call a CTC response. And you can see the prognosis of that last group is horrible, and changing therapy didn't matter. These patients have fundamentally chemotherapy-resistant disease. So Erin Cobain from the University of Michigan, who is a rising superstar in our field, has been trying to design a trial called BioCheck, in which we would basically do the same thing, but give these patients something besides chemotherapy that doesn't work for them. This is 1/4 of all patients with first-line metastatic chemotherapy in the breast cancer setting. So this was our initial design. After a few years, it finally went to the NCTN Breast Cancer Steering Committee and sadly was disapproved about 2 weeks ago. But Erin is quite resilient, and we're going to bounce back from this and figure out what we can do so that we don't keep giving these patients therapy that doesn't work. So in conclusions, my 25-year journey, I've conducted and published several stamp collections leading to my and others getting promoted. I'm pleased with that. My and others getting funded. My wife has been pleased with that. We've never missed a meal, to my knowledge. My mom and dad were quite proud of me. They both died of or with cancer a decade ago, but they thought I was dead. Although my father was a businessman, and after my 3rd move, and my brother worked for Eli Lilly. He's a doctor too, but he worked for Lilly making drugs, and my dad asked me what was wrong, why couldn't I hold a job and be like my brother? And I said, I think I'm doing all right. Anyway, I have yet to accomplish any correlative science study in the cooperative group that's definitely changed practice, but I have participated in studies that have led to new prospective styles that are designed to change practice, and I hope that's a good model. And we've identified interesting biological phenomena that may or may not be important. I think they are, and I think we can build on these. So I have a few pearls here. Think like a scientist and think like a doctor. Ask yourself, so what, after every study? Stamp collecting's okay, it can lead to new hypotheses and get you published, but ultimately we're here to mail letters. And thank you, Lee. A bad tumor biomarker is as bad as a bad drug. You can't just say, oh, it's a tumor marker, who cares? We care a lot now. And leave your ego at the door. This is all about our patients, not about us. And have patience. It takes a long time to get things done in the cooperative group, but it's worth it when we do. And have fun. This is really a great group of people to work with. So having said that, I just want to, again, history. On your far left is Jim Holland, who started the CLGB. He was the first guy to write the first protocol of combination chemotherapy in ALL. Dr. Fry, who's standing next to him, by the way, that's Jim's wife, of course, Jimmy Holland. Dr. Fry and Dr. Fryrick often get credit because they ran the protocol, but Dr. Holland actually sent me the protocol with a date on it. It was before those 2 guys got to the NCI. They were all good friends, but they were all competitive. Sadly, Dr. Holland passed away about a month ago. We miss him dearly, of course, and of course, Jimmy Holland passed away on Christmas Eve. Ross McIntyre and Rich Shilsky were chairs of CLGB during my time there, were terrific supporters, and these are just other people, including our own Ed Liu, who, like me, was smart enough to get out of CLGB before they changed the name and came to SWOG, so we're pleased with that. And then these are people in SWOG who have been very supportive of the things we've been trying to do, and I think you'll recognize most of them as you go through here. But in particular, I'm going to point out Alistair Thompson and Andy Godwin, who will be taking my place tomorrow at about 8:45 to run the Translational Medicine Committee. We're in good hands. I'm really excited about having them come aboard. And on your far lower right is myself and a guy much shorter than I am, but better looking. And we were receiving, being inducted into the FASCO Fellows that day, so we're both quite proud of that. And I'll end with this. Many of you wonder, why do I hang out with Jimmy Ray? And I've asked myself that many times. One is because he's quite successful. He got a collegiate professorship. I had to tie his tie for him, but that was... The second is I've been trying to teach Jimmy, he's just a PhD, and what MDs actually do on Wednesday afternoons, right? We take off and we get 18 holes in. And the third is Jimmy is actually known as a chick magnet, as you can see here. But frankly, that's his beautiful wife, Jan, in the middle, and those are her friends or my colleagues, Lisa Newman, at a fundraiser. So Jimmy, you got lucky and you married up. Congratulations. Having said all that, thank all of you for putting up with me. Thank you for giving me the honor and the pleasure to serve you. It's been a wonderful 17 years, and I'm not going to die. I plan to keep coming back, and I'll be sitting with Gabe Hortabaji in the back of the room and just listening to what everybody else is doing. Thanks, Gabe. Thank you very much.
Right there. So, here's more. Yeah, everybody, stay up, yeah.
That'll be on Twitter.
So we're going to,
When
go
I
ahead.
got elected as president, the ASCO staff taught me how to use Twitter, and I said, I don't want to do that. And so you have to. So now I do Twitter all the time. On Twitter.
I know, you're good at retweeting. We're going to break tradition. We're not going to have any questions or answers at this time, but we are going to do something a little special.
So Dan, SWOG, we'd just like to thank you for your 16 years of selfless service to the breast committee as the TM co-chair. I'm pleased to know that you're not going away, you're just stepping back and letting the new people take the leadership, and you're leaving us in really good hands here. I won't go into the science that you've championed. You've done a wonderful job describing that. Instead, I would just like to mention that you've been an incredible champion for the young investigators, and it's quite evident by your slides here. You're showing you're very proud of your trainees and your mentors, and we are...
Helen, who was your first paper written with? Dan Hayes. Helen was one of our fellows at Georgetown, did a great job, and we wrote a paper on a drug that didn't work, but it was still a good paper.
There's a lot of them.
And you've also done a wonderful job of bringing the PhDs in. And for me, it was incredible to learn that. When I was first met Dan at Georgetown, I was always a little bit leery of MDs, and I'm not really sure exactly what goes on in the clinical trials. And it's just been incredible for me professionally to understand clinical trials and translational medicine. You've been wonderful for that. You've been a wonderful role model as how to be a mentor. When I think about my own trainees, I think, what would Dan Hayes do in this situation? Just an all-around good guy. Definitely not a role model when it comes to golfing. The reason I'm still horrible at golf is because Dan keeps giving me pointers, and I'm awful. So, in honor of you, we, SWOG, have gone to great expense to produce this commemorative plaque. Come up here for your office. As you can see here, Dan, is it J or F?
It's actually F, but that's okay.
Okay, well, you like to edit and all my grammar and typos and everything, so that is for you, Dr. Hayes.
very
The photos.
Very good.
But because I'm also being trained and mentored by Dr. Ellis and Dr. Blanky, I thought I should also provide you with something a little bit nicer for your office. This is in honor of your dedicated service to SWOG. Thank you very much for all you've done for all of us and for all of your patients. I get a picture of you and Lee with us?
Yeah.
While we're doing our pictures of ourselves, everybody can go learn something and make a difference in the lives of patients with cancer. So we thank
there's
you
that,
all, and we will see you at the next plenary session.
Picture of you guys.